Good morning, everyone, and welcome back to the second session of the fourth day of the CCMB Open Week. Uh, today, now we'll be having a session on the animal cell culture, and it's a product for today, and there are prospects for tomorrow. This session is going to be conducted by Dr. Avtar Singh. He's the current in charge of the tissue culture facility at CCMB, and he has more than 7 years of experience in handling animal cell cultures. And let's hear from him.
Hello, am I audible? Okay. Good morning. Today I am going to present some of the things that we are okay that we are working in CCMB Animal Cell Culture Facility, and the title of our today presentation is Animal Cell Culture. That we are making a product for today, but that will be useful for the tomorrow or in the future. So before starting, I just would like to include some basic concept. What is cell culture? So cell culture is a process by which cells are grown under okay. Cells are grown under uh, tight controlled conditions, and generally outside of natural environment. And these cells are we can get it from normal cells, like normal tissues or organs from animals or human, and then we grow those cells outside of their body, and usually we grow in dishes or flask, and then we provide them nutrients and other kinds of factors so that they can grow, and then they utilize the most of their available resources at that time. And as soon as they grow, they are taking more space and they are become more confluent, and confluent is like they cannot grow after that confluency, and then we need to passes those cells. So those cells which we isolate from uh, human or tissue or organs, those are called as primary cells. So as soon as we do subculture, those cells will become a cell line, and if we provide some kind of pressure, just either introduction of some kind of plasmid or do some changes, then those cells will become cell stains, but they do have some limited lifespan, so they die after some time, some period of time. But we can immortalize these cell line, normal cell line, and they become immortalized cell line, that they do have infinite multiple passages, so they you can grow as many as time you want. And cancer cells, which is already our transformed cells, so they are automatically immortalized cell lines, most of the cell line. And what are needed for cell culture, like this is the about how cells are grown, but what are the essential things we need to do cell culture? So first of all, we need a cell line. So cell line, either you can produce or there are available everywhere, and CCMB is also providing cell line, and these are some of the common cell line that we used for our experiments, and from the different species, different tissue origin. For example, MCF7s are from breast cancer cell line, HeLa cervical, H129 and lung cancer, like that. And just to grow all these cells, we need to have some kind of food that is called as medium, and this medium will provide all essential nutrients, vitamins, inorganic salt, and FBS, that is fetal bovine serum, is also added to the cells, so that they can get more of macromolecules or hormones to grow in those conditions. And in addition, we also antibiotics, just to get rid of all contamination such as bacteria or fungus or yeast. And as soon as you grow cells, you need to preserve those cells, because sometimes when you grow cells, if you don't need for particular period of time, for example 1 year, so that you can freeze those cells and you can keep it at in -70 refrigerator, or usually we do it in liquid nitrogen, that is under the -130 degrees Celsius though. So the, if you need in like after 5 years or 10 years, you can revive those cells just for future prospects. And sometimes when we are growing cells, there are time when contamination uh, comes in your culture, so we have to get rid of those contamination, either by treating with antibiotics for bacteria or fungus or fungi. And culture condition are necessary for growing all these cells, so that we can we use CO2 incubators, provide CO2, and we provide 100% humidity, and other factors such as temperature. We almost most of the cell lines are can be grown at 37 degrees Celsius. And based off morphology, these are the different morphology that is are existing right now. This is fibroblast. They are more of bipolar and multipolar structure, elongated in shape, and they can attach on if substrate is provided. And there is another one is epithelial kind of structure, which is more of um, uh, more of attached cells. And lymphoblast cells are more of rounded cells, and they don't need any support material. They can grow in suspension. And these are this is one of the flasks that we are showing, that we usually grow our cells in cell culture flask. And these are the settings of cell culture. These are our staff who are working in tissue culture facility. So some of the staffs are working in biosafety cabinet, where we do cell culture, because we cannot do outside, just to get avoid contamination. So these are the biosafety cabinets. And these are CO2 incubators, where we keep our cells to grow. And on the back side, you see there are liquid nitrogen tanks, where we freeze our cells just to preserve those cells. If anyone need it, we usually provide or do experiment as per requirement. And this is the microscope that is routinely used by every other lab. Why culture cells? So that is a major question, why we culture cells. So there are different reason for that. First is we can study model system. So is there any basic biology of the cells, how cells interact, how they are behaving in a disease condition, and how drug can affect those cells. And sometimes we do toxicity testing also, so whether is there any drug which is available in the market, whether it has any toxic effect, or it is beneficial for the those cells to kill. And we also study in this cancer research. So we study how normal cells are different with the cancer, and how that cancer cells, they have altered cell signaling. And we also study these cells in virology to make more virus depending on our interest, so that we can develop vaccine against those virus. We also do genetic engineering to changes into in such a way that we can make more protein out of it. And gene therapy, sometimes we use these cells in gene therapy to replace a non-functional gene with the functional gene. And what we do in tissue culture facility, we provide training to individual who join in CCMB, and provide cell line to CCMB researcher and outside as well, as per MT agreement. And these are the training that we have provided in last some of the years. In addition, we also authenticate, because sometimes what happen when people are working on cell line, and when they are working, they get mixed cell line if they are working with more number of cells at a given time. So, or if we are growing for a longer time, then there are changes in genetic makeup. So in that case, we have to figure it out whether we are dealing with the right cell line or not. So that we authenticate cell line with using STR profiling, that is a short tandem repeats profiling. These are small repeats present in the cells and also known as microsatellites. And based on that, there are 8 core STR markers are available that we usually do PCR against those. And then we compare with the available database of those particular cell line. And this length of str are from 1 to 16 base pair, and we use multiplex pcr to identify those uh, amplicons. So this is just a generalized view. We usually take cell line, extract DNA from cells, and then we PCR amplified from DNA, and then separate by capillary electrophoresis, and then convert these PCR fragment into the allele so that we can create our database, and then compare with the available database whether those cell line are authentic and cell line have the same source that we were expecting. In addition, we in our tissue culture we establishing a model for organoids. So it is very important to understand what is organoid, because in the last session with Pavitra's lab, you have uh, listen about more of organoid things. So this is a general factor that we usually get organoids from adult cell, either from somatic cell or fibroblast, and then we make it into induced pluripotent stem cells by introducing these Yamanaka factors. Those are already studied, and these factors are essential for changing into induced pluripotent stem cells. And based on what we need, so that we can add additional factors to create a different kind of germ lines, that is mesoderm, endoderm, and ectoderm, and that we can confirm with the markers whether we are getting the same kind of uh, genotype or phenotype. And then based on if we add more cocktail of specific lineage, then we can generate organoid for respective organ. So here just an example of brain organoids. Uh, this is a, because pluripotent stem cells are used to differentiate into specific lineage, such as a neural lineage here to form a neural tube, that is neuroepithelial buds. And upon addition, further addition of specific cocktails, they can be induced to form mini brains. And these organoids, which is called as organoid, and these organoids faithfully recapitulate fetal brain development, and the cortical layers are very well preserved in these organoids. In addition, we are also we have already developed these hepatic organoids from iPSCs with addition of different substrate or different uh, media and some chemicals. And then we have confirmed these with the specific marker for those particular organs. So for example, here alpha fetoprotein, cytokeratin 18, 19, these are elevated in 18 days human organoid compared to the iPSCs, which does not have any um, which does not they lack these proteins. And even we have also confirmed with western blot analysis. Here you can see this OCT4 is a Yamanaka factor, which is usually present in induced pluripotent stem cells, which is further down in organoid. And these organoids show more of cytokeratin 18, albumin, and alpha fetoprotein, that is a marker for liver. So these are the application of organoid. We can study disease mechanism, and these are useful in regenerative medicine. And we can do targeting, editing, drug efficacy test, and study infectious disease such as COVID-19 as well. And this is useful in the development and stem cell biology. And we can make a biobanking of patient-derived organoids, and we can study host microbe interaction. And metabolomic, transcriptomic, and proteomic analysis can be done by using organoids. And we also do localization study. So there are two ways you can study localize how protein localize. So there is if protein is already present endogenously, we can stain with the two different protein with the antibodies, and then secondary antibody with different colors. So for example, here these are stained with green fluorescent protein, and this is red. And if so if they are localized, you can see there is a more of changes of that green and red to yellow color. In overexpression study, if you want to study where it is that protein, if you overexpress protein by introducing some transfection of plasmid into the cells, then you can study localization study just putting a red fluorescence protein just after your protein of interest. So here just an example, you see there is a more of this is nucleus, these are nucleus and these are the cytoplasm. This protein is present in the cytoplasm, more potentially more in the cytoplasm, but as soon as it is treated with some treatment, you can see there is a localization of this cytoplasmic protein into the nucleus. And this is where protein is already inside the nucleus, so upon treatment there was no difference. So you can study localization by the either by staining for endogenous protein or overexpressed plasmid, and then see how that plasmid making protein, and then it goes into different subcellular compartments. And we also making a monoclonal antibody, which is a well established model to produce hybridoma technology. Hybridoma technology is a well-established method to produce monoclonal antibody that is specific to antigen of interest. So in this case, we usually take antigen, that is, we need to raise an antibody against it, and we immunize the mice with these antigens, and thereafter we isolate B cells from the spleen, and then we fuse these isolated B cells with the myeloma cells, which have a greater potential to proliferate indefinitely, and then we make hybridoma. So in this hybridoma, after fusing myeloma cells and B cells, this hybridoma will have a mixture of cells, which is either B cells or myeloma cells, and combination of B cells and myeloma cells. So then we select these hybridoma in the het medium, that is hypoxanthine, aminoterine, and thymidine media, in which myeloma cells will die because they don't have HGPRT enzyme, and B cells will automatically die after some time. So whoever cells which is myeloma cells along with B cells, they can survive in het media, that is because they carry enzyme of HGPRT from B cells into the myeloma cells, and then we propagate these hybridoma cells and harvest antibody either in vitro or in vivo. So this technique was invented by Cecil Milston and Collers in 1975, and they shared the Nobel Prize. And it is a useful method to produce identical antibodies in large quantity. So there are many applications of monoclonal antibody, which is in therapeutic application in cancer, cardiovascular disease, infectious disease, inflammatory disease. And for example, cetuximab or panitumumab are available antibodies, monoclonal antibody, which target EGFR to suppress the growth of cancer. And these antibodies are helpful in the prevention, diagnosis, and treatment of disease. And in the diagnostic application, we can use this antibody for ELISA, microarray, biosensor, immunoblot for particular protein of interest. And in the last, we also do in cell facility heterokaryon. So this model of heterokaryon is mostly basically similar kind of hybridoma technology, in which a donor cell which is having a RBP, GFP expressing uh, proteins, which is fused with the recipient cells which is having mCherry, and then mix and seed on cover slip along with the treatment of cyclohexamide, so that no new protein will be synthesized. And then we fix an image. If there is a shuttling from donor cell of the nucleus to the recipient, then we can visualize under microscope. So in this example here, you see these cells, which is we have fused NIH3T cells with C3G cells expressing CHA cell expressing C3G protein, and then we combine these two. So here we are studying, so you can see these nucleus which is having C3G protein, and upon treatment the C3G translocate into the recipient nucleus of the cells, that is NIH3T3 cells. So overall, these are the things we do in cell culture. And thank you, and I will accept any question or any suggestions from your side. Thank you.
We will now take a few questions. Mr. BVV Padasadi and D Padasadi will be answering questions on YouTube, and we'll ask a few questions to Mr. Avtas. So our first question is, what causes cell surface proteins to promote cell-cell contact? By Siddharth.
What was the question?
What causes cell surface proteins to promote cell-cell contact?
So, so there are some junctional proteins which are basically interact with the cell-cell contact. Like for example, tight junction proteins, and like in the nucleus, they transport whatever they specific for their junctional localization. So they transport those protein to the epithelial of the cells, and then they contact with the other cells.
What is the difference between the protocol of plant tissue culture and animal tissue culture? From Pratiti.
Plant tissue culture. Can I answer?
So the question was, what is the difference between the protocol of plant tissue culture and animal tissue culture?
Look at yours.
Will you answer, ma'am, this question?
Can you come?
ఇక్కడ రండి మేడం ఆన్సర్ చేయడానికి plant and animal cells have plant and animal cells have very different properties with respect to their growth outside of the systems. When you're growing animal cells outside of the system, they require not only nutrients but a specific set of growth factors. And primary difference between animal cell proliferation and plant cell proliferation is there's a limit on the number of times an animal cell can proliferate. That's called a Hayflick limit. Whereas plant cells can give rise to any tissue type. All of them are pluripotent, so you can grow them in tissue culture for any number of divisions, unlike animal cells. For animal cells to grow outside the body continuously, they have to undergo a change called immortalization. Is that clear?
So further questions will be
Yes.
answered by Dr. Virad. Why do we use cell lines? By Vishwa.
Cell lines have enabled us to understand a variety of properties of cells in our body and in any kind of animal systems. Okay, fundamental scientific understanding has come about quite a bit only after, cell biological understanding has come up particularly in the last 70 years after we achieved the ability to culture cells outside the body. So the advantage of a cell line is that unlike primary cells, which will stop dividing after a few divisions, cell lines can be immortally grown in culture. That means they can go through any number of passages. You can freeze and you can also revive them. And how pathogens act on our cells, how our cells respond when there is a pathogen, what happens if you expose our cells to a particular kind of drug. The pharmaceutical industry has considerably depended on testing a variety of compounds on cell lines before they have been taken up for clinical trials and animal studies. And there are many other uses, like all our vaccines, they have to go through a cell line exposure before they are tested in any other context.
Mr. Amrish Rai, please unmute yourself to ask your question.
Hello, ma'am.
Yes, tell me.
I just want to know that is there are some cell lines present
Yes.
to study the neurodegenerative disorders.
Exactly. Yes. Yes.
So what are those cell lines? I want to know.
See, you can use actually the neuronal cells where you are studying neuronal degeneration. Okay.
Mm-hmm.
Or
Mm-hmm.
you can actually make a model of degeneration by using other cell types and introducing those molecules that cause neurodegeneration. For example, some molecules like form aggregates like Huntington's proteins. Okay.
Yeah.
So if you make these aggregates in a non-neuronal cell line also, some pathways required to understand neurodegeneration can be studied. Ideally, it is best to use cells that have the properties of neurons.
So, okay, someone wants to study pharmacological study for
Yes,
neurodegeneration.
yes,
So,
yes,
he or she can use cell lines or not? Means,
exactly. Yes, they can. So you can generate a cell line which is constitutively expressing the proteins that form these aggregates. Okay.
okay. So,
So you can use those cell lines and then subject them to any kind of downstream effector functions. You can use them to test the efficacy of drugs. You can use them to
yeah.
study fundamental pathways that are causing cell death because of the aggregation. Okay. And it
So,
need not be restricted to neuronal cells. See, you can have degeneration of muscle cells. So whichever
yeah.
context, ideally you would prefer to use cells of containing having those properties and that background. But in the
So,
absence of those, you can also mimic it using some other cell lines.
so means we can generate some knockout or knockdown cell lines for those
Yes,
studies.
surely. And you can maintain them in culture for a long period of time.
Okay, okay, ma'am.
If
Thank you.
if if that gene
Yeah.
is not essential
Mm-hmm.
for
Yeah.
survival. Okay.
Yeah.
Uh, Thank you, everyone. And this is the end of this session. Stay tuned for the next session, which is about microscopy and its use in biology. Please fill the feedback form. The link will stay active for an hour. Thank you, Dr. Avtar Singh and Dr. Radha.